I wish
even
it could
through
be in person, and I could
absolutely,
that would be great.
absolutely. This will be just a first time, then we'll do it live. Thank you so much for accepting.
Oh, of course, my pleasure. You know, that's what I really miss during this whole shutdown. Is this? I miss the interaction with people and share results and get feedback, and it's just
yes, absolutely.
And this is the best we can do, so let's do it.
Yeah, excellent. How are things over there in Boston?
Well, it's not looking good. We're in the midst of exponential growth of number of cases, and so it's pretty bad. We are a little bit outside of Boston, but even here, it's not good. So in the coming weeks, I guess it's the same everywhere right now in North America and everywhere in South America, where you are, in Europe, where my family is. They're all struggling. It's difficult.
Yeah.
Is your lab running, or are you guys open, or?
So it's so basically we are working in taking turns. So like two people tops in the lab. Yeah, working at the same time, and with very strict rules to enter and go, and to try not to mix people. So a bit like this. So it's really like, I don't know, maybe 10% of the work we normally do. Yeah, it's difficult.
I also say to people in my group, I mean, they're all worried about not being productive enough and not being able to do whatever they can. I said, look, it's fine. It's fine. We're going to be fine. Don't worry about that. Yes, everybody's going to be slowed down. It's fine. The lab is okay. You guys are doing great. Let's just get through this year and then catch up the slack again. But right now, just make sure everybody is safe. I must say, I'm very impressed by my lab in their spirit. They just keep going, and I think it's fantastic.
Yes, I think the same. My students are just working very hard, and whenever they can, they just...
I think it's very impressive. By the way, we have a few minutes. I just want to make sure my slides work, because
yes, please.
I did a presentation last week, and for some reason my slides weren't progressing till
yes, let's try it.
Make sure that works. Let's see. Can you see?
Yeah.
Let's try to make this happen. The most important thing is if I move to the next slide, can you see it moving?
So at the moment, I can see you are not in like full presentation mode, but I can see the different little slides, etc.
That's kind of strange. That happened to me before. That's why I want to do a quick test run.
Yeah. There you go. Now I can see the slide, the full slide.
Yeah, but does it move?
No, it's not.
That's the problem. I had the same problem last week, and I don't know why that is.
By any chance, you have two monitors?
I do have two monitors, but I have used it for 8 months, and it has always worked, and stopped working last week.
Oh.
So that's that's what I don't understand. I'll try it again. Okay, what do you see now? Because for me,
I can see how does the genome work and 2 meters, but
that's not the slide I'm on.
Yes, but it doesn't move.
That's so strange.
Yes, so there I can see the presentation with an alternative.
See.
I'm thinking, and okay, I can see the presentation now. Yes, now it's moving. Yes, now it's moving.
No, it's exactly the same as it was just now.
Yeah.
So it's moving?
Yes, it's moving.
What do you see now?
I see the first slide, chromosome folding by phase separation. The second one, I think. Yes, now it's working. Excellent.
It's a mystery to me.
Déjame.
Yes, let me introduce you to Dr. Felix Resillas, our director.
Nice to meet you.
Professor Dekker, it's a great honor for me to have you with us, at least in this way. And
yeah.
I used to be a formal postdoc at Gary Felsenfeld Laboratory. This is why we are here, as you can imagine. We are really excited about your talk, and
thank you
very much to give your time.
I really look forward to it. And when were you in Gary's lab? Because I haven't. Is this lab still open?
Yes. Gary, he has a surgery recently, but he's still working. He's still thinking. He's as you know, he's a marvelous person.
Yes.
We love him very much, and I'm keeping in touch with him. Of course, the lab is slowing down, but we have all his legacy. Many of us.
Yeah,
and Mayra used to be my PhD students not so long ago, but then we are CTCF kind of persons around.
I won't mention about CTCF too much today, unfortunately. I'll talk mostly about higher order structures, but Gary has done so much beautiful work.
Exactly.
He was really, one of the, when I started my postdoc in Nancy Kleckner's lab, I devoured all his papers because he really did all of the important early work, and later on continues to do important work on chromosome organization.
Sure.
And the other thing. And he's, as you said, he's a gentleman. He's a beautiful.
Yes, and as you certainly can imagine, I have been so proud to have the chance to spend several years in his lab, and it was a very nice time at that time. In fact, it was the beginning of the CTCF. I was there where he published, I was not a co-author, but he published the first CTCF paper in Cell with the characterization of the boundaries, the concept of boundaries.
Yeah, but he also did what I still think is not appreciated enough, the very important observation that a boundary can be separated in like a boundary function and installation.
Well, that's my paper. That was my paper.
Oh, that's your paper. That's such a. I mean, sorry, I did not remember who published it, but that is a paper. I keep reminding my group it's really important.
Yeah. In fact, the same happened with the IGF H19 boundary because there was only blocking activity, but not insulation.
Correct.
And then this is, in my opinion, this is still an open question. I mean, why and where is the difference between these two activities? In my opinion, it's still not clear.
No, I don't. For me, it's not clear either. And we're doing some more analyses on what you need to do to have insulation, and what you need to do to form a domain boundary. And those are sometimes conflated. Even in my early thinking about these things, I was probably naive myself. And
we were all naive.
Yeah, I mean, everything we do right now, we should realize everything we do now is naive. We will say that to each other 10 years from now. But I think we found some other DNA elements close to CTCF sites. And I would like to go back to some of these very well-characterized insulating boundaries, the one you studied, but also like in the chicken and globin locus, but also in H19. There are other sequence features around CTCF site that could play a role.
Yeah,
totally. And I know other people, I know Bing Ren has a nice paper, or at least story, I'm not sure it's published yet, where he finds that there are flanking sequences to CTCF that are very important for domain formation in addition to the CTCF binding and looping. And I think that is, there's a lot to be learned there still.
Yeah, there's a lot of room there still.
Oh yeah.
Well, one more time, thank you very much. We appreciate very much that you give your time to us, and we're expecting to hear a very nice talk. Thank you very much.
Great. Okay, so I think so people's gonna keep on joining the meeting, but I think I can start presenting. So really, it's a real honor for me to present today's speaker. Dekker is an investigator of the Howard Hughes Medical Institute and professor and co-director of the program in Systems Biology, Department of Biochemistry and Molecular Pharmacology at the University of Massachusetts Medical School. His laboratory studies how genomes are folded in 3 dimensions. He introduced the concept that matrices of chromatin contact frequencies can be used to determine the 3-dimensional structure of chromosomes. He then invented the chromosome conformation capture 3C technology to obtain such matrices and solved the first structure of a yeast chromosome in 2002. Since then, his group has pioneered development and application of a series of molecular and genomic and computational approaches, such as 5C and High-C, to map and analyze the 3-dimensional folding of genomes at KB resolution. His work has led to new insights into the internal organization of chromatin fibers, the formation of chromatin looping interactions involved in long-range gene regulation, the organization of the interface nucleus, the structure of metaphase chromosomes, and the general folding principles of complete genomes. His group also pioneered the use of 3D genome folding data for the novel genome assembly, which is a more extensively used tool nowadays. Dekker was awarded the Keck Foundation Distinguished Young Scholar in Biomedical Research Award in 2007, the ASBMB Young Investigator Award in 2011, the 2018 Biochemical Society International Award on the 2018 Genetics Society and the 2018 Genetics Society of America Nowitzki Prize. And in 2013, he was elected fellow of the American Association of the Advancement of Science. Thus, as you've heard, Professor Dekker is, to me, he's really who started and gave rise to the whole 3D genome organization field in which I work. So it's really fantastic for me to have him here today. We thank him truly for accepting our invitation. And without further ado, Professor, go ahead.
Thank you very much for that very kind introduction. It really is my pleasure to speak to all of you today. I wish I could meet all of you in person, spend maybe some time at your institution, but I'm sure we can find opportunities in the future to do that. It's great to meet you all, and what I want to do today is present some pretty recent work from my group on the larger scale organization of chromosomes. And the title includes the term phase separation. I apologize, this is a very popular term right now. I don't try to popularize the science I'm doing, but I really believe phase separation does play a role in chromosome folding. In fact, it's a pretty old idea that I want to explain to you in the first part of the talk. And in the second part of the talk, I will explore areas that I think actually are somewhat understudied in 3D genome folding, and that is related to the topological state of chromatin. So in general, let me know the slide is transitioning. Myra, did it go to the next slide?
Yes, all good.
So excited. I had some problems with my slides recently. Okay, so in general, the problem that we're trying to understand is how chromosomes fold. Now, why are we interested in that? If you look at the human genome, which is an incredibly long molecule, it's about 2 meters, which is about my height. I can tell you it's tall. What the field, the genomics field, has focused on for many years is trying to understand everything that's inside that genome. What is encoded in the genome? The genes, regulatory elements, and so on. And from these kinds of studies, we were hoping to learn how genes are regulated, how the human genome actually works. But it became pretty clear many years ago now, actually, even during the Human Genome Sequencing Project, that in order to understand how the genome actually works, we have to understand how that sequence is organized in three-dimensional space. And as Myra already introduced, we have developed over the years a variety of genomic molecular based technologies to probe the three-dimensional folding of chromosomes. Of course, we're also using imaging, which is incredibly important. And actually, I must say, I suspect that most innovation in genome folding for the coming years will be in the area of imaging and live cell imaging of dynamics of chromosomes. But what we have done over the last many years, actually, is to employ chromosome conformation capture-based methodologies to probe chromosome folding. So I know many of you are very familiar with this, but just briefly touch on the concept, because most of the data, not all, most of the data I will show you today is based on these kinds of assays. So what we do in these kinds of assays is to measure which pairs of loci inside the nucleus are physically very close together, so close that we can cross-link them with formaldehyde. And we do this genome-wide. So across the genome, we'll map which sequences are near each other in three-dimensional space. And from such contact data, we can learn about chromosome folding. So traditionally, what we do is we start with cells. These are typically intact cells. You can even do it in tissues that are being fixed. So we're fixing chemically all the loci that are near each other in three-dimensional space. And then through a series of enzymological steps, where we first fragment chromatin with a restriction enzyme, and then religate interacting pairs to form these ligation products that represent physical interactions. So what we really do in this experiment is we convert a very difficult problem, knowing which sequences are near each other in three-dimensional space, to a much easier problem, and that's just analyzing DNA sequences to identify these ligation products that tell us that two loci are interacting with each other. Now, over the years, we developed a variety of variants to this method. Today, I will mostly talk about Hi-C, which is a genome-wide variant that allows us to make genome-wide contact maps to identify which sequences across the genome are near each other in three-dimensional space. Okay, so the main thing that I want to focus on today is actually the spatial compartmentalization of chromosomes, as you can see that during interphase. In fact, this is the traditional compartmentalization of euchromatin and heterochromatin, known for many, many years. And one of the first features we discovered, or rediscovered, so to speak, when we performed Hi-C on cells. So here you have a Hi-C contact map, and I just want to give you an example. This is chromosome 14 in human cells. I believe this is a lymphoblast cell line. What you typically do when we get Hi-C data, we plot these contact maps where we plot interactions, or we on the axis of this heat map, we have one chromosome from left to right and from top to bottom. And each pixel in this contact map represents a pairwise interactions between the two corresponding loci. Now, many of you, of course, have seen these maps. You typically get this very strong diagonal for loci that are relatively close to each other in linear genome. But when the first time we make these maps, I'm thinking this was probably 2007, what was striking to us immediately was this pattern, this flat pattern or checkerboard pattern all across the chromosome. Now, at the time, we already already explored this to some extent, and I'll talk much more about it today, about how do these structures form. When we look at these maps, we can see, as I highlight here in the blue lines, these are in this area here off the diagonal. This is a pretty frequent interaction between two domains. Each of these domains can be several megabases, and they can be tens of megabases apart in the chromosome. These two domains interact with each other, but they don't interact with everything else. There are certain domains they don't interact with, which are the white spaces. So one way to interpret this is that what we did, and but now is a pretty common way of looking at this type of data, is that a chromosome is composed of a series of domains that I indicate here in circles or ovals on top and to the left of the contact map. And you get this checkerboard pattern if you imagine that there are two types of such domains that I color red and blue here. They're two different types of domains, and these alternate along the chromosome, and they interact in three-dimensional space, such that all the red domains interact with red domains, but red domains don't interact with blue domains. Blue domains interact with blue domains. So it's a simple pattern like that of alternating types of chromatin domains with different properties. And if they self-associate, like water interact with water and oil would interact with oil, you end up with this checkerboard of patterns. And in fact, you can simulate these things by polymer simulations, and it reproduces these kinds of contact maps quite nicely. So what we think this checkerboard pattern really means is that each chromosome, even though in linear space they alternate between red and blue domains, in three-dimensional space, all the red domains come together and all the blue domains come together. So the chromosome is basically moving back and forth between these, what we call compartments. Now, if you look inside, what is inside those domains, for instance, looking at gene expression or the presence of active regulatory elements and so on, we've quickly discovered that, for instance, all the inactive chromatin in a given cell type is in the blue compartment, what we call a B compartment, and all the active chromatin is in the red compartment, A compartment. So this is actually the classical separation, spatial separation of heterochromatin and euchromatin as seen by Hughes. This is one of the first features seen by Hughes, but still we know very little about how these structures form. Now, some of you probably use this type of data to look at much higher resolution contact maps, for instance, looking at loops between CTCF sites. They all happen along the very dark diagonal. There's a tremendous amount of work being done at that level of structure. I won't talk about that today. I really want to focus on these very large compartments and the mechanisms by which they separate from each other. And so, because as I said, it's one of the first structures to be observed by Hughes, but we still know very little about how this happens. We have seen this, of course, in the microscope. Heterochromatin is separate from euchromatin. Why is it and how does it work? Now, a current mechanism that people are proposing that could lead to such spatial separation is phase separation. I just mentioned here a number of groups. I probably miss a few, I apologize. A number of groups that have proposed mechanism based on phase separation that can lead to this checkerboard pattern of interactions. And some of them have done beautiful work to show that polymer simulations where you have, for instance, you put in attractive forces between the red domains and you put attractive forces between the blue domains, you can simulate a system like that and it will form contact maps similar to what we see in Hughes. It is one thing to simulate an attractive force between red domains or blue domains. But as a molecular biologist or a biologist in general, like myself, I want to know what are the molecules? Why does euchromatin attract other euchromatin? Why do they become separate from heterochromatin? These are things we really don't know, even though the theory suggests it might be phase separation, like water and oil are separating from each other. What is actually the molecular mechanism? So why is this hard to understand or to study? So we've gotten really interested in those mechanisms. And again, as I said, the checkerboard represents spatial clustering of chromatin. The idea is that this happens by phase separation because red domain attracts red domains, blue domains attract blue domains. There might be, the black lines here indicate there might be some forces or proteins that attract or self-assemble to bring these domains together. But what are these proteins? Now, one problem with these kinds of contact maps, even though I think they look beautiful, they have problems and they have limitations, is that it's a static population average contact map. If you give me a map like this, I wouldn't be able to tell you in advance which domains are inactive or active. They look very similar. Their attractions seem equally strong. Wherever we are on the checkerboard, the checkers look similarly dark. So I can't really say much about whether there are strong or weak interactions and whether they are different for A and B compartments. So that those kinds of questions led us to ask, can we measure inside cells the attractive forces that will bring heterochromatin together or that will bring euchromatin together? Can we actually measure it? Again, by high C, we actually cannot measure it. We just can observe the final separated state, but we can't really say much else. So this led actually us to start a project, which at the time was very courageous, because these were kind of like, it was kind of an abstract notion of what we were trying to do. And it was actually quite difficult, but this was led by really a super talented student, graduate student in my lab, Huda Balaksal. She recently graduated, who teamed up with Tyler Borman, another graduate student in my neighbor's lab from the bioinformatics department. And he did a lot of the computational work I will show you in a second. And then Denis and Andrew, John and Sergey helped us with some follow-up experiments, as well as some of the polymer theory that I had to learn as we were going along. And in fact, I mentioned here at the bottom, this work had been in bioRxiv since 2019. It just got accepted in Nature Genetics. And we always complain about reviewers. As scientists, we like to complain about reviewers, people who review our work. I want to give a big shout out to the reviewers for this paper. I don't know who they are, but they did they gave us tremendous suggestions to improve the paper. We did all of them, which is why it took us some time. They really improved the paper, and I think this was just really, I'm so thankful for what they did. The story got so much better. Okay, so let me introduce to you the concept of how we're going to measure the attractive forces between heterochromatin and euchromatin. So I said to you that phase separation is one mechanism by which A and B compartments can spatially segregate from each other. The idea is actually quite simple, and I think intuitively not so hard. If you think about a chromosome, here on the left, I just draw it as little balls, a string connected balls of A's and B's. And each A block and each B block is actually composed of smaller beads. It could be just a 10 kb locus. The A and B compartments each are typically a megabase or so these are pretty big. And they alternate along the linear chromosome. Now, in three-dimensional space, as I indicated, the A's will attract A's and the B will attract B's, so that the A and B basically become separated from each other, as I draw here in the middle. Now, this happens if A domains interact with other A domains, and B domains like to interact with B domains. And that's the forces with the dotted lines here. They are somehow attracting each other. And we want to measure that attraction. How attractive is a B to a B and an A to an A? Now, this is where some of the polymer theory comes in. And this is why it's been great working with people like John Marco, who helped us a lot in our thinking. A polymer like this, or a chromosome like this, can form A and B compartments like shown in the middle when two conditions are met. First, each of these blocks of A or B needs to be sufficiently long. Second, the attractive forces between them need to be sufficiently strong. Okay, so you can show that here in a phase diagram. If you can either have large blocks, so the A strings and B strings are very long. When those blocks are long enough and the preferential interactions between A's and B's are strong enough, you become phase segregated, like in this dark part of the phase diagram. If you have very short blocks, so for instance, instead of having 1, 2, 3, 4, whatever, 10 or so of these A beads strung together to form one block, if you just have A and B's monomers alternate, like one A bead and then one B bead and so on, then the block is very short. A system like that, or a chromosome like that, will only form this phase segregated checkerboard if the attractive forces are really strong. Then they will phase separate. If the blocks are very large, like I drew here on the left, that system will phase segregate even when their attractive interactions are weak. Okay, so it's kind of like each monomer in these blocks like to interact maybe with one other one, but if you string them together, you get some kind of a cooperative effect. Okay. So if you look at a real chromosome, here is chromosome 2, here's a high C map. We can see the checkerboard, which means A and B blocks are spatially segregated from each other. It means in the phase diagram of phase segregation, we're somewhere in that gray area. But we actually don't know where. Now remember, what we actually want to measure is how strong does B like to interact with B and A likes to interact with A. So we'd like to know where are we on the x-axis of this phase diagram. Now if we zoom in here, so we know the chromosomes are segregated. A and B compartments are segregated. So we're in the gray area, but we don't know what interaction strength we're actually having. It could be very weak, could be very strong. It all depends on the relationship to size of the blocks. Now, in chromosomes, the compartments are several megabases in size. Each A and B compartment is several megabases. So the block size is large. We're somewhere here in the phase diagram, which means the interaction strength could be very weak. That would lead to phase separation. Could be very strong. That would lead to phase separation. We really don't know. So this was kind of the theoretical picture we had. Chromosomes are segregated, but the blocks are very large. And now I don't know what the interaction strength is. And that led us to the following very simple experimental approach to measure the interaction strength. And that was very simple. What would happen if you reduce the block size? So if we in situ fragment chromosomes into smaller and smaller pieces, the block size will become smaller and we're dropping down in the phase diagram. And at some point, there will be a point that we can no longer phase separate. And that point will tell us what the interaction strength is. So it's actually theoretically straightforward, I think, and experimentally approachable. So that's what Huda actually started doing. And it took her quite some time because typically when I say things are very simple in the lab, it actually takes a lot more effort to actually get it to work. So what she did is she started with isolated nuclei, in this case from K562 cells. We chose that cell line because it has lots of epigenetic data from the ENCODE project. And we start soaking in a restriction enzyme. So now we're cutting the chromosome in smaller and smaller pieces. Now, if you have a chromosome conformation, like here in the bottom example, that is held together by lots and lots of strong interactions, even if you cut the DNA, that structure will not fall apart. If you have a structure that has very few attractive interactions, if you cut the DNA, it will just dissolve. Okay. Now, so the idea is very simple. We're gonna take nuclei. We're going to in situ fragment this chromatin with restriction enzymes. This is all before we cross-link DNA. We cut it in pieces and ask how long does it take to dissolve structures, like an A compartment or a B compartment. We wait some time, then we fix the material and do high C. Okay, so what happened? Did certain structures or different parts of the checkerboard disappear or not? And then we did a whole series of experiments to measure various things. I will just highlight a few in today's presentation. Okay, we call this high C on liquefied chromatin because what happens if you fragment the chromatin becomes somewhat like a liquid, as I'll show you in a second. And I'll hope to convince you today that when you do these kinds of experiments, we can measure chromatin interaction dynamics and the general forces that bring A and B compartments into their segregated state. So what happens if you do this with nuclei? Here we have an example. Here I show you an isolated nucleus, and I stain the DNA with DAPI, and it looks like a purified nucleus. Now, if you soak in HinD3, this is supposed to cut the chromatin quite frequently, but not that frequently. In fact, in chromatin, HinD3 digestion generally produces fragments that are about 20 kilobase in size. So in fact, we know in this condition, the entire genome is cut in 20 kb pieces. If you then do imaging, and you look at a microscope, not much changes. And maybe a little, the pattern of the DAPI looks a little bit different, but it still looks like a nucleus. Now, what happens if you cut the DPN2? This is supposed to cut the chromatin quite frequently, but not that frequently. In fact, in chromatin, HinD3 digestion generally produces fragments that are about 20 kilobase in size. So in fact, we know in this condition, the entire genome is cut in 20 kb pieces. If you then do imaging, you look at a microscope, not much changes. And maybe a little, the pattern of the DAPI looks a little bit different, but it still looks like a nucleus. Now, what happens if you cut the DPN2? In this case, we're cutting the DNA in something like 3 to 6 kilobase fragments. The whole genome is cut in small fragments. Now we see what we call liquefied chromatin. You see these beautiful droplets of chromatin budding or blabbing from the nucleus. They don't really come out of the nucleus. There seems to be still a membrane, nuclear membrane around these droplets of chromatin. There's no lamina, so it kind of tears the nucleus, but it protrudes from the nucleus but doesn't get outside. Actually, it's a classic view that you can also see in apoptotic cells. Remember, apoptotic cells also cleave their DNA. So this is already a remarkable result in my view. You can cut the entire genome in 20 kb pieces and the nucleus seems to be intact. If you cut it in smaller pieces, now chromatin becomes like a liquid. Now, what happens if you do high C on these nuclei? So here we have a control nucleus, and we see the checkerboard that again represents compartmentalized A and B compartments. Remarkably, I think it's still absolutely stunning in my view, if you fragment the nucleus at 20 kb fragments, the whole genome cut in 20 kb fragments, nothing changes by high C. So those 20 kb fragments just stay where they are. They have plenty of interactions with other fragments so that they're kind of like frozen in this matrix of interactions. Remarkable, I think. In fact, we've now done an experiment where we take these nuclei and just wait for a month. But even after a month, these nuclei are not changed. Now, if you cut the whole genome with DPN2, so now we're looking at 6 kb fragments, now we see much more changes. Now we can see the checkerboard changes. We went from A and B checkers that alternate with 2 alternating patterns in control and in 3 digested nuclei. When you cut with DPN2, it seems like only one of these patterns is still there. The other one is much weaker. Okay. So from this, we can already conclude, this is actually from a physical point of view, really interesting. There must be a stable contact with another fragment on average every 20 kb, at least once every 20 kb in the genome. And that holds the whole genome together as a completely connected network of interactions. If you cut it in smaller pieces, fragment starts to move, but it seems like that's not true for both A and B compartments. It seems like one compartment pattern is still preserved, the other one is gone. So we wanted to calculate some way of quantifying that this actually happens. How can we calculate whether a given locus across a chromosome has lost interactions? So one way we thought about doing this, it's maybe not the best, but we thought it was a useful way of calculating this. On the right here, I show you a nucleus, and in green there are 2 chromosomes. I can't remember which chromosome this is. This is a picture from Wendy Bickmore's one. If you have a locus here by a red dot, it typically interacts mostly with other loci on the same chromosome. So typically when you do high C experiments, you see loci mostly interacting with other loci on the same chromosome. Now, if chromatin becomes liquefied, you cut the chromatin in small pieces, that locus can start floating around the nucleus and interact with other chromosomes. So when a locus becomes soluble or becomes dislodged from its chromosome territory, it will it will have reduced interactions in cis, but increased interactions in trans. So we can actually measure this in high C. So we can actually ask, okay, we are a given locus in a control nucleus has so many cis interactions and so many trans interactions. If I now fragment it with DPN2, the locus starts moving. And now it has more trans interactions than cis interactions. So we use those numbers to calculate something that we call loss of structure, because it indicates that a given locus has lost its association with its normal neighbors and now is free to float through the nucleus. And we can calculate that very easily. So here we have along chromosome 2. I show you here red and blue the A and B compartments again. I used to show them as red and blue circles. You can do principal mode analysis to actually quickly identify the positions of these A and B compartments, just when the value is negative or in the B compartment. When it's positive, it's in the A compartment. It's a widely used method for calling these compartments along chromosomes based on high-C data. So this is the typical pattern of A and B compartments along the linear chromosome. And now we measure if you fragment the chromatin with DPN2 and loci start to move around, we can calculate that as I just explained by an increase in trans interactions. And you can see that the pattern of mobility of these loci after fragmentation mirrors the compartment profile. Loci in the A compartment tend to be more mobile than loci that are in the B compartment. They move much less. So it already seems from this initial plot that in the A compartment, there are far fewer interactions holding loci in place, so that when you fragment the chromatin a little, no, DPN2, loci quickly start to move around. Whereas in heterochromatin, when you cut to the same extent, these loci don't move away because they are still interacting with other loci. As if in the B compartment, there are just more interactions per kilobase than in the A compartment. Now, I mentioned in passing almost, assuming that we're cutting equally frequently in A and B compartments when we do DPN2 digestion. And of course, that is not obvious. In fact, the A compartment tends to be more accessible in general. When we do ATAC-seq or DNase 1 mapping, typically the A compartment is more accessible than the B compartment. So we checked that actually. You can fragment the chromatin with DPN2 and just sequence the ends to identify where we actually digested the chromatin. And when you do that, you see there's quite some variation along the chromosome. There is some correlation with compartments as well. So now that we knew where we were cutting and how frequently, you can actually normalize for that and take that into consideration and correct this blue plot for the cutting efficiency to obtain this black plot, which is corrected for digestion. For slight biases in digestion efficiency, you end up with a plot that looks a bit more noisy, but we still see the same thing. Loci in the A compartment are more mobile than loci in the B compartment. Okay, and this is unrelated to the fact that we have a slight bias for digestion in the A compartment. Okay, so in general, what we learned from these experiments is that if you cut in 20 kb fragments, nothing moves, because there are sufficient number of interactions for these 20 kb fragments to keep them in place in the nucleus. If you cut in 3 to 6 kb fragments, now the A compartment becomes much more fluid than the B compartment, suggesting there are more interactions holding B together per kb than the A compartment. But that was just one time point. We just digested the chromatin for a few hours and did high C. What happens if you do a time course? Can we actually measure the kinetics of this process in much more detail? So that's what Huda did in this experiment. She started at nuclei, added DPN2. Again, this is all before fixation. And then took samples at 5 minutes, 1 hour, 2 hour, 3 hour, 4 hour, or overnight, 16 hours. That's a little bit too long, actually. And what you can see is that digestion is basically complete after 1 hour. Fragments are 3 to 6 kb, and it basically stays like that for quite a while. The data I showed you up to now was actually this 4 hour time point. But now we can do high C at these different time points. So here we look to see at control nuclei, at the same as what I showed you before. Beautiful checkerboard, A and B compartments are segregated. But then at 5 minutes, nothing happens. Even at 1 hour, look at this. 1 hour, the chromatin is completely digested in 3 to 6 kb fragments, but the checkerboard is still intact. We have to wait longer, 2 hours, 3 hours, and here's the 4 hour time point I just showed you. And then 16 hours looks almost completely liquefied, although you can still see one of the two checkers is still present, and I can tell you that's the B compartment. So it seems that the fragmentation itself happens within an hour, but then this dissolution, the loci start moving around, actually takes several hours. So even though I say the chromatin is liquefied, it seems to be quite viscous, and loci move very slowly. But it allows us to measure the real dissociation kinetics, like how much time does it take for a locus to dissociate from its A or B compartment and start moving around so that it can interact with other chromosomes. Now, in order to address that more kinetic question, what we next did is calculate the same metric I explained to you, this loss of structure metric, that tell us how many loci of a given bin or position across the genome have moved away from its original chromosome. And you can do that for each time point. So at each time point, so at t is 0, there's little change. But then as time progresses, we can see more and more of the loci become mobile. This loss of structure becomes larger and larger, and A compartments move faster than B compartments. That also means that for every locus, we now can measure the half-life of its interactions in cis. So for instance, here over time on the x-axis, on the y-axis, we have how much the locus has become dislodged from its native structure. And we can see we get a beautiful dissociation curve. And from this, we can easily fit the time point at which 50% is solubilized. And that we call the half-life of that particular locus, the half-life of its interactions in cis. So we can really measure a dissociation rate here. So we go from measuring contact maps to now measuring the dissociation rates of contacts. And we can measure that again across the genome. So now instead of measuring compartment status or something like that as a static track, we now have a profile along the chromosome of chromatin interaction dynamics, the intrinsic dynamics of a locus. If you were to cut that locus in small piece, how long does it take for it to move away? And again, same as what I showed you before, except now it's opposite. Loci in the B compartment have long half-lives. Their interactions are stable. Loci in the A compartment have short half-lives. They dissociate quickly, about 50 minutes, so it's relatively quick. And loci in the B compartment typically take about 100 minutes. So it's about 15-minute time difference. Again, we can correct for differential or weak biases in digestion efficiency, but it really doesn't affect the results. Loci in the A compartment have short half-lives in their interactions. Loci in the B compartment have longer half-lives. So to go back to that original picture I showed you, from a static High-C map, we learned that A and B compartments are spatially segregated, and we assumed there were attractive forces between the red domains and between the blue domains, but we didn't know whether they were strong or weak. Are they equally strong for A or B, and so on. What we learned from these experiments that I just outlined to you is that now we know that in the B compartment, we have more interactions holding the B compartments together than in the A compartment. Or at least, if there are more equal numbers of interactions holding these domains together, in the A compartment, those interactions have a high dissociation. They can quickly dissociate. The only reason in cells that this situation is actually spatially segregated is because there are many A's strung together to form large blocks of A's and B's, so that even though there are weak interactions between them, in the end, just like Velcro, in the end, put all together, they interact very strongly. But intrinsically, in the A compartment, interactions are much more dynamic than in the B compartment. This, of course, might not be too surprising. Heterochromatin, we all think, is more compact, more hold together by maybe HP1 proteins and other types of proteins. And the A chromatin, active chromatin, is more open. Now, the power of this fact that we now have this genome-wide track of chromatin interaction dynamics for each locus across the genome, we know its intrinsic chromatin interaction dynamics. We can go beyond just looking at A and B compartments. We can ask, how does that relate to the local chromatin state, its histomodification status, and so on? So this is one reason why we chose to look at K562 cells, because these cells have so much epigenetic data made by the ENCODE project and others, that we can now look at every locus and relate it to a whole variety of chromatin features. For instance, if we look at loci that are associated with nuclear speckles, these are subnuclear bodies enriched in splicing factors. Loci associated with nuclear speckles are enriched in highly unstable half-lives. Very few of them have stable half-lives. On the other end of the spectrum, loci associated with nuclear lamina, they tend to have very long half-lives. Interactions at the nuclear lamina are very stable. And then in between, we have some really interesting findings. Polycom. So polycom is another inactive chromatin compartment in the nucleus. This is silent chromatin. It's bound by a variety of factors that people have analyzed and identified loci associated with polycom. If you look at loci associated with polycom proteins, they are quite unstable. The polycom domain might be quite stable because it's long, but each locus inside the polycom domain is actually engaged in relatively dynamic and unstable interactions. If you look at HP1 proteins, these are heterochromatin proteins. There are three kinds of them: gamma, beta, and alpha. Or H3K9 trimethylation, which is bound by these HP1 proteins. H3K9 methylated chromatin tends to be very stable. It has stable interactions. It's condensed heterochromatin. But the three HP1 type of proteins have very different chromatin interaction dynamics. Beta is intermediate, alpha is more stable, but chromatin associated with HP1 gamma is very unstable. And again, loci at the nucleolus are also relatively stable and so on. So this is just an early analysis of these type of data that we now have, but we can start to relate chromatin interaction stability, how stable are the interactions, to local chromatin state. And that is going to give us a handle on the molecular mechanisms of why certain interactions happen between certain chromatin domains. For instance, if you look at the polycom subunits, we now know that polycom is actually much more diverse. There's not just one polycom state, there are several, and they depend on which polycom proteins bind there, which subunits are there. And you can see that some polycom complexes containing SUSY12 or NF2, BMI1, they're intermediate stability, but when the CBX8 subunit is present, it's much less, less stable. So there's even diversity in stability within the polycom bound chromatin. So this is going to give us a list of candidates for proteins that could mediate chromatin interaction stabilities and attractions between A and B compartments. Okay, so this was really just a very technical story of a new method we developed to study chromatin interaction dynamics. So initially, when we did Hi-C, we get a static map, as I indicate with this diagram here, of the folded nucleus, the folded genome inside the nucleus. And we know inside we have A and B compartments. But now by using this liquid chromatin Hi-C, this new method we developed, we can color this interaction map based on interaction stability. So loci at the periphery here in dark red, they have much more stable interactions, but loci like near transcription sites or nucleus speckles, they're very unstable interactions around those loci. But at the polycom domains, these form beautiful bodies inside some cell types. We can see intermediate levels of chromatin interaction stability. So again, we now have a much more dynamic view of chromatin interactions in A and B compartments, and we think the compartmentalization between A's and B's really is possibly driven by the B compartment because they have very strong self-attractive forces, and these interactions are very stable. Okay, so we are really planning to use this assay to start to study the molecular players that bring A's together and B's together and the factors that determine the stability of these interactions. I think this is actually an important approach because we need to go beyond these static maps and start looking much more at chromatin dynamics. Okay, so I have one other aspect of these A and B compartments that I would like to share with you today. And that is what actually happens when two B domains that are separate along the linear chromosome, they come together because of these attractions between the B's. When two of these B domains come together, and as I've said several times, we think this is related to phase separation, do they fully mix the two chromatin domains, or do they just touch? Typically, when people think about phase separation, and people do beautiful in vitro experiments with chromatin fragments, and they all form these beautiful droplets of phase-separated chromatin. They actually can mix to some extent. And of course, in liquid phase separation, two droplets of water, when you put them together, they will merge and they form one droplet. What happens with chromatin inside cells? The way I draw it here, they're not very mixed. They're near each other, but not necessarily very mixed. So we're starting to be very interested at these interfaces. What happens when these domains come together? Do they mix or not? If you look at chromosomes, so not even at compartments, but look at chromosomes. Again, I borrow some beautiful pictures from Wendy Bickmore. I usually take pictures from her lab because the imaging is so beautiful. If you look, or from Anna Palmbo's lab for that matter, if you look at two chromosomes, they don't mix very much. Here in red and green, we have two chromosomes. They're very near each other inside the nucleus, but they don't necessarily completely mix. So we've gotten interested in those kinds of mixing events, either between chromosomes or between compartment domains. There's definitely extensive contact between chromosomes. We can measure that by Hi-C, and Anna has, of course, studied this extensively, showing that there's definitely some interdigitation between the chromosomes. So we got really interested in measuring this. So where chromosomes or compartment domains interact, do they mix? Do they become entangled? And I mentioned topo 2 here because one has to imagine if you put two chromosomes near each other, like two balls of yarn, they don't necessarily mix very easily. You probably need some topo 2 activity, but topo 2 is very abundant. So is at their interface where two chromosomes touch, do they become mixed or entangled or not? So the top scenario is when there is no entanglement, no mixing. The two chromosomes in blue and brown here, they are near each other, but they don't really mix. When you have topo 2 activity present and lots of entanglements, you can get lots of mixing between these chromosomes. It's hard to know, based on Hi-C, for instance, to know whether it's the top or the bottom scenario. Okay, so we got, thinking about this, we got really interested in this topic of entanglement and mixing of chromosomes. So any of these mixing or entanglements can be a problem for the cell. For instance, during mitosis, chromosomes, of course, have to become disentangled and separated from each other. But biologists always say this is not a real problem because there's so much topo 2 in a cell, it can either entangle it or disentangle it, and the cell will figure it out. But it turns out we actually don't know much about how much mixing or entanglements between or within chromosomes exist. And one reason we don't know is because it's very difficult to analyze. So for instance, I show you here on the left, a black sequence from one chromosome and a gray from a different chromosome. Here, they're not entangled. They're just one is on top of the other. Or they are entangled or catenated, like on the right. And it's the difference between the left and the right structure is just the fact that they're just on top of each other in one case, and one is looping through the other in the other case. But by microscopy, they look almost identical. Even on this slide, to some of you, they might look identical, but they're not. What we almost serendipitously learned over the last several years is that we can actually learn about entanglement and mixing using multi-contact chromatin interaction data, not just pairwise data as you measure in Hi-C or 3C or 5C or Capture-C or any of these C methods. You can actually learn about the extent of mixing if you look at multi-contact. And this was a project that was initiated by Philippe, a computational scientist in my lab. He is now back in Europe, and later followed up by Davout, a physicist in my lab. Bastian and Sunny are two experimentalists who contributed data to this project. This paper just got published. Okay, so let me introduce you a little bit about multi-contact data. So typically, when we do 3C or Hi-C, we focus on pairwise interactions between two loci. But in reality, when we do these kinds of experiments, you actually get long strings of ligation products that represent multiple contacts that happen inside a single cell. So for instance, here on the top left, you can see a cluster of a red, a yellow, and a brown locus. They're all somehow interacting with each other. If you cross-link this and then fragment the chromatin and religate, as you do in a Hi-C experiment or a 3C experiment, you actually get a whole string of ligations. The red one gets ligated to the yellow one, gets ligated to the brown one. Normally, we would share this in pieces and only sequence pairwise interactions. But we can actually sequence this longer ligation string on a PacBio sequencer. You can sequence up to 10, 15 kilobase molecules, and we get like a whole string of ligation junctions. And these are interactions that are co-occurring in a single cell in a single subnuclear location. And here we call these CWOCs. Actually, this was Amos Tanai's lab who originally coined this phrase a few years ago. So what you then see when you map this to the genome, you can, for instance, see for this first example I gave you with a red, a yellow, and a brown locus, you can see red interacting with yellow, yellow interacting with brown, and so on. So we can measure these kinds of multi-contact strings of interactions. So what we did is, in this case, we looked at non-synchronous HeLa cultures, and we generated a couple 100,000 of these kinds of strings. The numbers are not as large as in Hi-C because PacBio can't sequence so many of these. Now, the first thing we asked, and other people have made this type of data, but they didn't necessarily ask this question, is the order of interactions in these strings random or not? If you have this first example I gave you about 3 loci all interacting simultaneously, and people have interpreted the data often in this way because this data has been used to see that a promoter can interact with 2 enhancers at the same time. So then you would get a 3-way interaction between the promoter, enhancer 1, and enhancer 2. If this is the type of structure we're capturing, you could ligate these ends in any random order. This particular example could produce a ligation string like this, the first one, or the second one, or the third one. They all actually represent the same structure. So the order of the fragments might be random in the ligation product. Or, and this is my favorite model, it's actually what we're capturing in these kinds of experiments is not a hub or a cluster of co-occurring interactions. In fact, what we're measuring is a path, where one fragment interacts with the second fragment, the second fragment interacts with the third fragment, and maybe the third fragment will interact with the fourth fragment. But the first fragment is not really interacting with the fourth fragment. So in that case, the order of the fragments inside the ligation path is not random. It follows this path through the nucleus. And in fact, that is what I think we're measuring. One way to distinguish these two things is to determine whether the order of fragments is random or not. How do we know that? One way to know that is if you look at the ligation string. Again, there are direct interactions, red to yellow and yellow to brown, and there are indirect interactions, red to brown. If the order is random, like this top model would suggest, then direct interactions should be equivalent to indirect interactions. You can just change the order and it shouldn't change anything. We have evidence to suggest that direct interactions are actually different from indirect interactions. So typically when we plot Hi-C data, we often plot interaction frequency between 2 loci as a function of genomic distance between them. And typically you see this inverse relationship. Loci that are very near each other in the genome interact frequently. Now this is true for direct interactions in this data, same as Hi-C. But indirect interactions actually have far fewer short-range interactions and more long-range interactions. This is on a log scale. So this is about 2 to 4-fold reduction in short-range interactions. So indirect interactions actually are different, quantitatively different from the direct ones. And that tells us that the order of fragments is not random. So we're not randomly ligating these cluster of fragments together. It's more likely we're following this path from one locus to the next to the next. It's basically like a 3-dimensional walk through the 3D genome as captured by this multi-contact data. We realized if that is what we're actually measuring, we can just walk along in 3-dimensional space and see what happens. So for instance, we can identify a walk that starts in one chromosome and through a number of steps walks into the territory of another chromosome. And maybe from those kinds of walks, we can learn something about the interface between these 2 chromosomes. Now, if the chromosome is very mixed at that interface, you can imagine if you follow a walk, you step from one locus to the next, you're gonna start at chromosome 1, you take a step, you end up on chromosome 2, you take another step, you might end back on chromosome 1. And in other words, you can step back and forth between the chromosomes quite frequently. You can be very mixed when it's entangled or not entangled. We'll get to that in a second. But if the interface between two chromosomes is very unmixed, they're very smooth surfaces between the chromosomes or between compartments, what will happen is very different. Now the walk will start in chromosome 1. It will take a step, it's still on chromosome 1. It will take a step, it's still on chromosome 1. And only once will it cross over, end up in chromosome 2. But once it's in chromosome 2, it will keep taking steps in chromosome 2. In other words, when you follow a walk that starts in one chromosome and ends in the other, when you have a mingled interface, the walk will go back and forth between the two chromosomes. When the interface is smooth, it will cross over to the other chromosome only once and then stay in that other chromosome. So what did we find when we did this experiment? So what I show you here is maybe a bit complicated, but on the x-axis is how long the walk is. And again, this is multi-contact data, and we have walks up to 10 interactions in one molecule. And on the y-axis is the frequency. Here we look at walks that start in one chromosome and end up in another chromosome. So there's always at least one interaction between the two chromosomes. Now what you can see is that even for chromosomes that, for walks that are very long, 5, 6 interactions, you typically have only 1 or 2 interactions between the chromosomes. In other words, on the right here, from short walks to very long walks, the number of interactions that just stay contained within the chromosome is actually quite large. For long walks, 80% of them are within the chromosome and only 20% go from one chromosome to the other. Okay, is that significantly enriched for interactions within chromosomes? So we do a simple permutation test where we permutate the order of fragments and ask the same question. And you can see each dot here is a permutation. That you can see that in all cases, we observe in experiment more intrachromosomal steps than expected by random chance. So that suggests that there's actually little mixing between the chromosomes, because otherwise we would go back and forth between the chromosomes very frequently, but in reality, we infrequently go from one chromosome to the other, more like what you would expect for these smooth interfaces between chromosomes. Okay, so we think we actually have very smooth interfaces between chromosomes, something that we didn't learn from high-C data, but we can learn from the multi-contact data. Now, it's the same thing happening with compartment domains. So we did this for 2 chromosomes, but of course we can do the same thing for a walk that starts in one compartment domain and ends up in another compartment domain. And when we do that, we get exactly the same result. I won't go through the whole slide, but in effect, we always see more interactions that stay within one of these blue spheres and rarely cross over to the other and then stay in the other sphere. So just like 2 chromosomes, 2 compartment domains don't mix very much. I told you they attract each other, but when they do, at their interface, there's limited mixing. They're smooth, relatively smooth interface. Now, finally, the very last 2 slides, what does it say actually about entanglements? Because why don't they mix? So here we did some polymer simulations of 2 domains, and they're closely held together. And it turns out if you take 2 chromosomal domains in a polymer simulation, you just put them together without topo 2 activity, there's no strand passage possible, the 2 domains actually will not mix. That's kind of counterintuitive maybe, but even 2 chromosomes or 2 chromatin domains compressed to each other, they won't easily mix. That only happens when you have topo 2 present. And in fact, Davoud could actually simulate a multi-contact experiment where you start a walk in one domain and you cross over to the other. You do a simulated multi-contact experiment in the computer and do the same types of analysis I just showed you for experimental data to show indeed that the experimental data suggests there's no mixing and no topo 2 exchange between these domains. So here we have a totally computer-based experiment, a simulation of 2 domains. We do a multi-contact simulation through this structure, and we can plot again the fraction of intrachromosomal steps as a function of the length of the walk, And compared to permutations, we always have way more interactions within the domains. And again, this is very similar to what we saw by experiment. In other words, the computer simulation without topo2 mixing reproduces the experiment. If you do have topo2 mediated mixing of chromatin, now the data look like random permutations of the interactions. So now we have complete mixing of the domains. Okay, so in summary, what we can actually measure is these walks through three-dimensional space. And by looking at walks that cross different chromosomes or different compartment domains, we see that their interfaces actually are not mixed. And we can explain that low level of mixing by a lack of topological entanglement. So this actually led us to a totally conclusion we didn't think of getting at, is that the genome is largely unentangled. So here I just show you an example of a polymer simulation of two chromosomes, each having two compartment domains, and they interact with each other. On the left, there are no entanglements. On the right, there are entanglements. They look quite similar, and by microscopy, they might look quite similar. But in fact, the multi-contact data allow us to show that at their interfaces, there's very little mixing, and there's no topological entanglement. Okay. Now, I just maybe open up for discussion. We don't know why topo2, which is so abundant inside the cell nucleus, doesn't just freely mix chromatin all the time. There's two ideas from John Marco's group and Davide Micheletto, who published two really exciting PNAS papers that showed that SMC or cohesin-mediated looping could actually steer topo2 to constantly disentangle chromatin. I won't have time to explain this, but there is a link actually back to cohesin-mediated loops that can actually help explain why at the chromosome level or the compartment level, chromosomes are disentangled. And that would happen if cohesin-mediated loop extrusion steers topo2 towards disentanglement. Okay, I went a little bit too long anyway, so I'll stop here. I'm more than happy to take any questions. I acknowledge all the people along the way and my funding agencies, and all of you for your attention.
Thank you very much. That was an amazing talk. So I'll just kickstart the questions with a couple of questions that I have. So when the data it's really interesting in the time course, the digestion time course that you showed us. I thought I could see some nucleosome bands in the overnight digestion. I don't know if these are nucleosomes or not, but I'm wondering if this liquefied state comes from getting at the nucleosome size range rather than just cutting more and more, but that maybe that's the unit to now be a liquid.
It's a very good question. There are definitely, you have a very good eye. There are indeed some nucleosome ladder that you can see in the later time points of fragmentation. It's a small fraction of the DNA, but it's definitely there. The average size of chromatin in the A compartment is about 3 kb, actually. And in the B compartment is about 3.1, I believe, or 3.2 kb. So it's slightly larger in B than in A. But the nucleosome fragment is not the dominant feature, but there are definitely fragments the size of single nucleosomes. I actually think, and I would like to explore this deeper, we do add the restriction enzyme DPN2 to fragment the chromatin, but I think we get the nucleosomal ladder by endogenous nucleases that are present in the nucleus and that that are active under the conditions that we use. We can even see some of that if you just take nuclei without DPN2 and just incubate them overnight. You can see very faint degradation into mononucleosomes and dinucleosomes, trinucleosomes. Those will definitely be the most liquefied, that's for sure. I don't think they drive the bulk of the result because it's a small fraction of DNA that is in that category. We're actually now starting to purify DNA from the smaller fractions to see where in the genome they actually come from, and are those loci particularly mobile?
Okay, thank you. Felix, go ahead.
Professor Dekker, one more time, thank you very much for your nice talk. And I have 3 questions very quickly. The first one is really intriguing, this delay in the DPN2 digestion. And I was wondering if we can explain this delay, since the whole genome is already digested by DPN2, that the delay is due to transcription factors, density and interaction, or in combination with the histone modification patterns. And this creates this kind of delay. This is my first question. The second question is related to this middle stage of stability in the polycom group of components. And my question there is, it doesn't have nothing to do, or some involvement of RNA. I mean, some kind of dynamics of the interaction between polycom and transcription of the genome, since we know that a lot of genome is transcribed, and I'm thinking in non-coding transcription. Yep. And finally, when you were talking at the end of your, when you were explaining about the topo 2 ideas at the end of your talk, it came to my mind those seminal works of Penman and the nuclear matrix, that one more time they were suggested that RNA and topo 2 may have some relation in the 3-dimensional structure of the nuclei. Then these are my 3 questions. Thank you very much.
Yeah, so the first question about the time delay between fragmenting and things actually dissolving. You know, I think it is related to transcription factors. We haven't spent a lot of time looking at transcription factors because the chip data is relatively sparse for that type of data. We focus mostly on histone modifications and things like that. We should go back looking at that because also for the HP1 proteins, the stability of heterochromatin mediated by the 3 different HP1 proteins, but also actually for the CBX proteins in polycom, seem to be directly related to the affinity of those proteins for the histone modifications. So it's almost, if that binding is very stable, the loci dissociate slowly. If the interaction is very dynamic, they move faster. Now, for instance, I'm very curious also about CTCF, because CTCF binds very stably to DNA, but also can form clusters. And they could definitely bring things together and hold things together. Now, there's another thing I didn't mention, but if you fragment chromatin, CTCF remains bound to the chromatin actually during this experiment, but cohesin dissociates. So cohesin gets off. So some of the interaction we're looking at might actually depend on cohesin also. Although at the 1 hour time point, most of the cohesin is off the chromatin actually. And then it still takes 2-3 hours for loci to dissociate. Now, what we're doing now, and maybe this addresses some of your questions, is that can we do chip or for some key factors over the time course and see, do they dissociate as chromatin becomes fragmented and liquefied? We actually don't know. We only looked for CTCF and cohesin in this case, and because CTCF remains bound, but cohesin gets off. But what about all the other factors? Another thing is just a technical issue that I am worried about. Is not worried. I'm worried about it enough that we do more experiments. We do this in purified nuclei. But if you purify nuclei, who knows what kind of factors you have lost. So now Denis, a really talented new student in my lab, is taking over this project, and he's already developed a method to do this in intact cells instead of doing it in purified nuclei. You have to do all kinds of tricks to get the restriction enzymes in, but it seems to be working. That can address some of these things. But I think we have to go back to identify the transcription factors or key factors that might mediate some of these interactions. Now, your second question was about polycom. Can you remind me what the second question was?
Yeah, that this is stable, middle stability that you show us can be due to some kind of dynamics of binding and reload dissociation of polycom due to genome non-coding transcription or general genome non-coding transcription?
I believe that is the case. So for us, it was really intriguing to see that the polycomb domain was relatively unstable. And that was kind of surprising to us. But there is all kinds of things happening inside polycomb. Polycomb chromatin, even though it's quite condensed, as you say, there are polymerases in there. There are non-coding transcripts in there. And also, as a biologist, I think it's good to have a structure be unstable if you want to control it. If you want to be able, if the cell wants to decide I have to activate this locus that used to be in polycomb, it needs to have some access to it. No? So typically when structures are metastable, it's also more regulatable. Now, does RNA play a role? So we did two things, and maybe you have better ideas. I would love to hear it. We did one experiment where we just removed all the RNA from the nucleus by RNAs. But again, some of the non-coding chromatin-bound RNAs, we might not be able to remove that way. But it didn't change much. Another experiment we did is block transcription. When we block all RNA pol II transcription, what happens? Not much happens, although some domains become more stable. As if transcription somehow made the locus unstable. Now, there was just one experiment we've done, but I think that points out to us that generally RNA maybe doesn't play such a big role, but for specific loci it may actually. And that when you activate it and transcribe it and maybe having the RNA in there could destabilize the locus.
Okay.
Now, one thing too that I want to point out is the intrinsic interactions inside polycomb domains are unstable, but the domain itself might be quite stable because if you look at it, at polycomb domains, they tend to be quite long, pretty large, like Hughes domains, they're tens of kb long. So their lengths may compensate for the fact that the interactions, each of them in the domain are weak, but because the domain is quite big, now it might be a stable domain.
Okay. Okay, thank you very much.
Great. So let's continue. So Andres Penagos.
Yes, thank you. Thank you for the talk. I think it's amazing. All this work on compartments. I was wondering, you found B compartment is the one that has stronger interactions. So I was wondering if maybe you could think of a biological state where maybe this can be inverted. Maybe you have now stronger A compartment interactions, like maybe cells with a very few heterochromatin or a lot of transcription, or maybe cells without lamins.
That's a good question. Yeah, so actually, that's a great question on this. Thank you. I think, so actually we're now applying this assay to a variety of cell types, starting with ES cells that have very relatively underdeveloped heterochromatin. We differentiate them and then we age them. And one thing that we have observed in other experiments is that compartmentalization is weak in pluripotent cells, gets much stronger in differentiated cells, and then gets very weak again in old cells. Old cells have problems maintaining heterochromatin. So we're going to apply this assay to see whether we can follow those kinds of, whether it's actually due to the fact that heterochromatin has strong or weak interactions in different states. We have not seen any conditions where the A compartment develops strong interactions. It's usually unstable in all cell types. It's really the B compartment that shows lots of development and differentiation in different cell types. So I don't know the answer yet in the S cells, where also the nuclear periphery is much less developed compared to differentiated cells. But your experiment to remove lamins is really interesting. I would love to try that experiment. We should try to do that. Actually, there are published cells that miss the lamin B receptor that is required for tethering heterochromatin to the nuclear periphery. So that could be a good cell type. But that's exactly the kind of approaches that we're thinking about doing. Remove by a mutant, or preferably, in my view, using an inducible Degron, use proteins, remove key factors in heterochromatin positioning in the nucleus or heterochromatin stability, and ask, if I remove this specific factor, how does it change the chromatin interaction dynamics for the B compartment? So yes, the question, of course, is, I have a wish list of 20 proteins. You can, of course, do only so many, but lamins, I will put it on top of the list. That's a really good one to start looking at. We haven't done these things yet.
Thank you very much.
Thank you for the question.
Next question, Brandon.
Yeah. Thank you very much for your talk. I also think that it was amazing. I apologize in advance if I didn't get the whole concept, but I have two questions. The first one is related to Dr. Resilla's question about polycomb. Mm-hmm. At the very first stage of your talk, I was thinking that the time the compartments needed to be digested was a consequence of chromatin accessibility. I was thinking about the physical accessibility for the enzyme to cut the chromatin. However, when you showed the polycomb compartment results, that doesn't seem to be the case, right? Because
yes, Brandon, that's a very good question. In fact, that was a concern of mine also, that some chromatin would just be accessible first, and they start dissolving earlier than other loci that get cut later. We did a tremendous amount of work, also thanks to the reviewers, to address that issue, and it doesn't seem to be the case. Surprisingly, actually, the way, of course, we overload this system with enzyme. We add a ridiculous amount. If you do a DNase 1 experiment or an aminase experiment to measure open chromatin, you actually use much milder conditions than we do. We actually come here with a sledgehammer and start smashing your genome. At the timescales we are looking at, it doesn't seem to matter. So actually, we repeated the entire analysis starting at the 1 hour time point, mm-hmm. when the whole genome actually is cut and there's no further cutting as far as we can tell. And if you say, that's my starting point, and now I measure the kinetics from then onwards, knowing that now everything is cut, we get the same results. So your question is really good, and it's something that kept me up at night for quite a while, being concerned about these kinds of caveats to our experiment. So you're absolutely right. That was a major concern. For some, the way we do the experiment here, it doesn't seem to matter. And you're right. As you also said for polycomb, we would have expected it to be cut later and appear stable, but in fact, it's the opposite. Mm-hmm. So that's a great question.
Okay, thank you very much.
You had a second question?
Yeah, I'm not sure about that.
Go ahead, ask it. Now is the time.
Go for it.
Well, I think this is about the very fundamentals of the concept. The way I imagine the molecules inside a liquid is that they are constantly moving. So I'm wondering how stable are the interactions you are recovering from your High-C experiments at the single cell level?
Well, I think when we're looking at unfragmented chromatin, intact nucleus, mm-hmm. it seems to be actually quite stable. Chromatin might move, but they might just move with their interactors.
Okay.
So that you're right, there is some movement there, and I was looking at some beautiful work from Alexandra Sidovsky. She is at NYU, and she measures the movements of chromatin by imaging, and you can see there's plenty of movement. So it's not completely a solid, but when you fragment it, now things become moving a lot, and they dissociate from their interactors, and now they start moving more. So again, that's a great question. Maybe I shouldn't say it's so black and white, liquefied or not. It's more gel to liquid.
Okay. Okay. Thank you very much.
Thank you, Brandon. Those are great questions.
Great. We have more questions if that's okay with you.
Yeah, absolutely.
Excellent. Amaury, go ahead.
Thank you. Thank you for this talk. I have a general question about this process. Do you think that this compartmentalization has a role as a transcriptional regulator, or is just a consequence of the transcriptional activity and the transcriptional factors or whatever is in there associated to these regions?
Yeah, that's a really good question. Also, because actually, you know, you touch on something that is really a big question in the field. We can measure all these things, but how does it relate to gene regulation? Like, and there's a big question about compartmentalization in general. Many organisms do it. Do you need it for correct transcriptional control? We don't know. One experiment that comes to mind now is another beautiful experiment from Wendy Bigmore's lab that shows that as cells exit mitosis, the nuclei become compartmentalized again, heterochromatin at the periphery. That is entirely transcription independent. The compartmentalization happens without transcription. It's driven by histone modifications. But those, of course, are related to transcription. To get them deposited in the right places often is related to transcription. So there is this chicken and egg situation, that you need the transcriptional profile of a cell to determine the histone modifications. There's feedback on transcription then, where the modifications can help reinforce transcriptional program, and that determines three-dimensional positioning. So, but what comes first and what is needed? We actually don't know. Well, why do you need to compartmentalize anyway? That's another problem I have. I don't know. One thing that we are currently, another postdoc in our lab is trying to do, is can we artificially place a locus in the incorrect compartment? And if you do that, does it affect its transcription? Now, this is another new experiment. People have done this, where they bring a locus to the nuclear periphery and then ask, does it change the expression status of that locus? And the results are mixed. Sometimes it does affect the transcription, sometimes it doesn't. So I don't know the answer to the question. One idea often is that bringing loci together of the same kind of chromatin type will create a subnuclear environment in which certain trans factors and regulatory factors become locally enriched, and that might help regulating genes. But then I talk to my colleagues who do live cell imaging of transcription factors as they move through the nucleus, and they tell me those factors can move anywhere freely at a very short time frame. There's lots of factor dynamics. Chromatin is relatively immobile, but proteins fit through the nucleus every time they have an opportunity. So anyway, this is a long way of saying it. I don't know, which is usually my answer to many questions, because there's so many things we don't know. But the link to function is the big gorilla in the room for a lot of chromosome biologists, including myself. We have these incredible tools in our hands to measure the structure of the chromosome, how it is dynamic, but how does it relate to gene regulation? And again, I think we need some new ideas here. So I would encourage all of you to come up with such ideas and try them out in the lab.
Thank you.
Thank you, Amari.
Abraham Roman.
Yes. Okay. Hello, Professor. Thank you very much for the amazing talk. I have a very general question. I was wondering what could be the role of this not mixing contacts between chromosomes for cellular health?
Yeah, I actually was surprised. I mean, again, we weren't really looking for it. We just stumbled on it, and we discovered that chromosomes are not mixed very much with each other, and within the chromosome, the compartment domains don't get very mixed. Why is that? I don't know, except that when cells go through mitosis, they will have to become disentangled from each other. So maybe they're just constantly getting their chromosome like in order and nicely organized and not mixed with other chromatin. It is surprising to me, and I don't know why this happens. I've been talking to physicists who study polymer mixing, and they have all kinds of ideas that this could actually be just an intrinsic property of polymers under certain conditions. But then as a biologist, I always counter and say there's topo2 mixing can happen quite easily. And in fact, when you add molecules in a tube and you add topo2, the whole thing will become mixed. That is not what's happening inside. The cell is working hard to have that not happen. Now, one thing I was talking to a colleague of mine who studies DNA replication, and he said that if you have replication forks are very sensitive to topological strain. So maybe any repair or replication or even transcription, which puts torsional strain on chromatin, becomes compromised when there's also entanglements. When the chromosomes are knotted and the polymerase has to run through a knot. How do you go through a knot? That's not so easy. So maybe that's what cohesin and loop extrusion actually is constantly doing. They're disentangling like little combs. They're just disentangling chromatin all the time so that other processes that act on the chromatin template, such as DNA polymerase, RNA polymerase, DNA repair, can occur relatively unobstructed. But again, I'm just waving my hands here because frankly, I don't know. We discovered this only a year ago or in the last year. And before that, I think my view was that there was plenty of mixing and that it was going to be not a big deal. But apparently, the cell is working hard to avoid that from happening. Now, we think currently that that cohesin, so there's this model here. Can you see my slide about SMC complexes? Right. So there's this beautiful 2 papers. I would encourage you to look at it, where if you have 2 molecules that are entangled, like here on the bottom, if you have loop extrusion by cohesin, it will actually extrude, say, on the gray molecule all the way till it hits this entanglement. So now topo2 knows exactly where to cleave to disentangle these 2 molecules. Excuse me. So they show beautiful simulations that if you have loop extrusion, which exclusively acts along one DNA molecule, it will actually, with topo2 present, it will always lead to disentanglement. So maybe, I mean, we all are interested in knowing why cohesin makes loops and extrudes loops, and CTCF maybe. So remember, CTCF sites are sites where topo2 binds as well. Maybe that whole machine of making loops and CTCF loops is all related to disentangling chromatin. So of course, now we're interested to see what happens if you have no loops, no cohesin. Do the chromosomes become entangled? And we don't know yet. But clearly, this is something that I think it goes back to the previous question. There are so many structural features of chromosomes we can analyze now, but relating them to function is always a challenge. I want to point out that just organizing chromosomes is important by itself. So a lot of the machines inside cells are just dealing with the fact that they have to organize all this DNA. And I sometimes like to think that cells are just, that by itself is a function. But who knows?
Thank you very much.
Thank you, Armand.
We have an extra 3 questions. I'll read them for you. From Itzel Hernandez, it is suggested that polycomb bodies cluster through phase separation, and recently it has been shown that these structures subtend different types of motion depending on the physiological context. In line with these results you presented, in line with the results you presented, how do you think these changes in motion are influenced, possibly by transcription factors interrupting polycomb long-range contacts? Moreover, I recently read that only cohesin but not CTCF lowers the interaction strength in polycomb domains. What do you think about these results?
Yeah, I'm familiar with most of those results. They're very exciting. To relate them to our observations of chromatin interaction stability is the way we calculate interaction stability at the moment makes it a bit difficult. to know whether two polycomb loci normally interact and then dissociate when there's transcription or they're disrupted in any way. We have to look more specifically at polycomb-polycomb interaction dynamics. Remember, the way we calculate it now is more general. We look at how a polycomb locus becomes dissociated from its chromosome. We should probably go back to our data and look more specifically at interactions between different polycomb protein bound domains and ask interactions between these specific polycomb domains. How do they change after fragmentation? We haven't done that, but that we should probably do, and then ask questions like what you relate to is what happens when there's transcription or cohesion mutants, and how do they affect interaction specifically between polycomb domain bound polycomb protein domain bound factors. And we haven't done that, so we need to look more precisely at polycomb domains in order to ascertain the effects of cohesion and things like that. But all these results do have in common is that polycomb interactions are clearly very dynamic and can be modulated by both transcription and cohesion. I think I'm particularly intrigued by the link between polycomb and cohesion, because we have also seen that we did some work with Bob Kingston's lab a while ago, where we could see that even if you have a polycomb bound domain, cohesion can just still get in there and extrude chromatin and form loops with other CD7 outside of the polycomb domain. So again, suggesting those polycomb domains are pretty accessible and pretty dynamic structures.
Great. So the next question is from Karina Jacome. She says, great talk, couple of questions. The genome is largely not entangled, but which could be the role of the few entanglements there are? In a diploid cell, the entanglement regions would be enriched between the same chromosomes?
Yeah, that's a very, very good point. Thank you for that question. It is true, there are still some entanglements. Actually, I went back, again, this is another great point brought up by a reviewer that said you should go back, and people have used single cell Hi-C and then reconstructed the three-dimensional structure of chromosomes. And if you look at those three-dimensional structures, do they have catenations or knots or entanglements? And we did that. We looked, I'm not sure it ended up in the discussion of the paper. Well, it may have. I should check it. But we did go back to those structures, and there are very few entanglements, but there are. Each chromosome can have like one entanglement over 100 megabase region. Our assays will never see that because we look at the population. It would be really interesting to know whether it would happen at specific sites. I have no idea, it could be. And how that relates to entanglements between homologous chromosomes. Yeah, that would be, it's possible. We haven't looked at that. We started looking at interactions between sister chromatids. Those are, of course, two chromosomes that after replication are entangled. After DNA replication, the sisters are completely wrapped around each other. They have to become disentangled during prophase. And that's the process I'm currently very much interested in. But we haven't considered any functional roles for the rare entanglements that are still there. They might be there, they might be important. I don't know. But it's a good point, and let me emphasize, our experiments show that entanglements are rare, very rare, but they are still going to be present at some places.
Yeah, and well, just a comment. It might be that, I mean, entanglements are important and also mediated by cohesin during recombination in meiosis, right? So there might be some link to that, even if we are in a somatic cell rather than.
Absolutely, absolutely, and I think there are all cytological observations of entanglements between homologous chromosomes and even between non-homologous chromosomes during meiosis. And there might be, and again, it links again to cohesin that might give directionality to entanglement and disentanglement. Yeah, we haven't looked at any of the other systems. Again, what I showed you today was just really our first exploration of these kinds of phenomena. But as you can see, as soon as you realize you can actually study mixing and entanglements, there are so many questions you can answer.
Yeah, absolutely. Okay, so one last question from Carlos M in the chat. The liquefied chromatin could damage the integrity of the nuclear membrane, or it hasn't been okay, so the liquefied.
I think the question, I see the question in the chat, is maybe related to damaging the membrane during the purification of nuclei.
Carlos M, if you're around, can you clarify? I think he means if there's a difference between if the liquefied chromatin could damage the nuclear membrane. That's the way I understand it, but I'm not sure.
If when you isolate the nuclei, you see a difference between nuclei with liquefied chromatin and those without. Like you have less nuclei with liquefied chromatin.
Well, when they purify the nuclei initially, Carlos, the nuclei look intact, most all of them, and then we add, but they're permeable enough that when you add a restriction enzyme, that restriction enzyme will get into the nucleus. Now it can go through little holes in the nuclear envelope that occurred during the purification, or they just get through the nuclear pores. Restriction enzymes tend to be pretty small, and they can go through the nuclear pores. When you do that, you start seeing the chromatin becomes more liquid-like. You see those droplets of chromatin, but they seem to be not getting out of the nucleus. It's more like a little bud on the surface of the nucleus. We know there's no lamina. The stained for lamina, there's no nuclear lamina, but there is probably a membrane, which is why the surface of that droplet is so smooth, because DNA wouldn't just float away outside the nucleus. Now, there might definitely be damage to the nuclear membrane under these conditions. And that might lead to loss of some protein factors that could influence the results. So that is something we're very cognizant of, and that's why we're going to do these experiments now in intact or at least permeabilized cells, but not purified nuclei. When you purify nuclei, the way we do it is you have to douse the cells and lyse the cells, and you have to go push that material through a gradient to purify nuclei. So it's actually pretty harsh, and it might damage the nuclear envelope. But I think before or after liquefaction, the chromatin, the nuclear envelope itself doesn't change much. There are going to be some holes in it, but I don't think it's going to be damaged to a significant extent. So much so that we're losing chromatin from the nucleus.
Okay.
Does that answer your question?
Thank you.
Thank you, Carlos.
There's another one. Do you have still a bit more time? I think.
Yeah.
Excellent.
I blocked off my afternoon to talk to all of you.
Oh, fantastic. From Ayerim Esquivel. In Drosophila, where there are transvection phenomena like contacts between homologous chromosomes, there are actually trans contacts, but when upon sequencing, I guess they look like cis contacts. My question is if there is a way to distinguish these types of trans contact between different chromosomes and between homologous chromosomes. I mean, if using these techniques, these contacts maybe should see like, if you see those as non-entangled.
Yeah, that is really a fantastic question. So you're absolutely right that there are trans contacts between homologous chromosomes. In high C, they look like cis interactions, and we can't really tell them apart. It's even actually worse in the cells we look at. The K562 cells is basically triploid, so they have 3 chromosomes that are all the same. There is a way out of this, and I want to point you to a paper from Ting Wu's lab. She published this paper last year, I think. We were a co-author on it, but only because we helped a little bit in the beginning. It's really her lab's paper. But Ting has been studying transfection in Drosophila for many, many years. And she figured out a way to use high C to distinguish between the homologs. Other people have done that too, using SNPs that distinguish the paternal and maternal chromosomes. So then you need quite a high density of these SNPs to enable to have enough information to tell for each interaction whether it's between two loci on the same chromosome or two loci on two homologous chromosomes. But anyway, she could do that in flies, and actually also in mouse. And what you can see is you can actually see in Drosophila where the homologs are paired to facilitate this process that you refer to, transfection, and you can actually measure that. And it's a nice, really nice pair of papers that I encourage you to look at, where she could find that the homologs are really tightly held together in some parts, but not so tightly in other parts. I hadn't thought of this, but your questions now encouraged me to think about whether we could study entanglement between the homologs in these kinds of scenarios. We should be able to do that in flies when we can tell the homologs apart, or in F1 hybrid mice, where you can make crosses between different mice strains so you can tell the homologs apart. It would be really great to do that. We should do that. I haven't done it, but I think it's a great idea. It's really a great idea. We should look at that in flies. Thank you for that. But that's a very good point. It's another limitation, not just in our work that I showed today, but in general in High-C studies, that in many cell lines we're not distinguishing homologs. So what we call cis interactions could also be some interactions between the homologous chromosomes. So that's actually generally a very good point.
Yeah. However, if you look at chromosome territories, you normally find the homologs.
Yes, you're right.
Fairly distant from each other. No?
Yeah, but in flies that's very different.
Yeah, exactly.
So in some organisms, that there's definitely pairing. But you're right. In mammals, often when people look at by fish, the chromosome territories, the two homologs. You're right, Marion. They're often not quite close together in any particular way.
Okay. There's another question. I think this is the last one. And the impairment in this entanglement of chromosomes could contribute to the formation of balanced translocations between chromosomes.
It could. It's certainly one idea that one reason you need to get rid of all these entanglements is that because if you have an entanglement that survives all the way till metaphase, anaphase, you could get a break, a coordinated break between the two chromosomes, and that could lead to a balanced translocation. So that definitely happens. Whether that is related to entanglement, I really don't know. Those balanced translocations don't happen very often. So it's going to be hard to know that when they happen, there happen to be right at the right moment an entanglement between those two chromosomes. One prediction of your question would be, I guess, that if you have a scenario when there's way more entanglement, say we find a mutant where the chromosomes, when the cell is not able to keep the chromosomes disentangled, now we have way more entanglements. Does that lead to more translocations? That would be actually pretty amazing if that were the case. That would also give a reason why cells work so hard to keep their chromosomes disentangled. But yeah, there could definitely be a link between these kinds of entanglements and genome stability.
All right. So, well, one last comment from me then. In the results you showed us about the different regions with epigenetic features having different strengths in their contacts. Sometimes I wonder if it's just the periphery and the nucleolus, which are like the most important features organizing all the rest. So heterochromatin binding the periphery plus the nucleolus and maybe the heterochromatin around the nucleolus as well. Just thinking about your data, which shows that these are the regions that are really like having interactions are really strong. I don't know.
Yes.
What.
Yeah, I agree with you. I think increasingly, that's why I got really interested in heterochromatin right now, because I think heterochromatin is the major driver for nuclear organization. And maybe it's almost a reason why we have so much heterochromatin in our genomes. Now, not in all cell types is heterochromatin always at the periphery versus the nucleolus. There's some cell types where it's the other way around. For instance, go back to an earlier question, to get rid of the lamins, if you get rid of lamin B receptor, all the heterochromatin moves away from the periphery and now forms this inverted nucleus where all the heterochromatin is in the center of the nucleus. This is fantastic work by Irina Solvay's lab. And I think, actually, I would love to do liquid chromatin high C on those inverted nuclei to see whether anything changes. But yeah, that is a very dominant feature in nuclear organization, is these stable interactions between heterochromatin at the periphery and heterochromatin at the nucleolus. Now, my colleague Paul Kaufman, who studies loci near the nuclear periphery and uses some method developed by others earlier, tells me every time I speak with him, he reminds me that not all the chromatin at the nucleolus is heterochromatin. There's also some specific active chromatin domains there. So it's a little bit more complicated than that, meaning being at the nucleolus or being at the periphery is not sufficient for chromatin interaction stability, but they are strongly correlated. But yeah, I mean, it makes me wonder just how important the heterochromatin in general is for nuclear organization.
Okay, well, thank you so much. we stole your time for quite a...
I enjoyed it very, very much. Thank you all for listening and for the great discussion, which I always enjoy the most.
Many students in the chat are saying, come soon and visit whenever possible. That would be...
Believe me, I will, as soon as I can. We'd love to visit. As some of you may know, I love to travel everywhere I go, I try to find as many species of bird. So I'd love to go to Mexico and come visit you guys and take the opportunity to visit some wildlife and of course have the great food and see all of you.
Excellent. All right. Well, with this, we close our seminar. Thank you very much.
Thank you for the invitation.
And thank you to everyone, especially the students. And we see each other next week. Nos vemos. Buen fin de semana. See you.
Bye-bye.
Bye-bye.
Bye.